So thank you very much. Thank you so much to the Jung Foundation and also to the board. It's a great honor to give this lecture, and also thank you to Blanche for hosting us here at the medical faculty. So our lab has been interested, as a long-standing interest actually, in the central factor in the central dogma of molecular biology, and this is messenger RNA. And this is the go-between that transfers information, genetic information stored in our DNA to the machineries that make proteins. And while DNA, as you know, is a permanent feature of our cells, it stays with our cells until the cells die, both RNA and proteins are transient. They do their job, and they are degraded. And in fact, at any time, in any cells, the amount of RNAs or transcripts and proteins you have is really due to the balance between the rate with which they've been synthesized and the rate with which they are degraded. But degradation serves also more general other type of roles. Degradation is used to remove molecules that are defective. And they can be defective because of problems that have happened in the way to make them. These are very complex biosynthetic pathways, or problems that come upstream from the DNA, from genetic mutations. Now, degradation of these molecules in the cells is carried out by molecular machines. And I like to make the parallel between machines that we have, or we used to have maybe in the office. Now we don't print anymore. But when we used to have in the office, these were machines, shredding machines, which would be used to eliminate documents that either are not useful anymore, or documents that would have dangerous content that would be dangerous if it would leak out. And in the cell, we similarly have kind of molecular machines which recognize these molecules, and they break them down. And they recognize molecules that are not normally needed, not needed anymore, or molecules that are defective. Now, like when your machine in the office breaks, you have to understand why it breaks down to be able to fix it. And it's not just important to know what is the part list of these machines. You have to know how all these pieces come together to try and understand if one piece breaks or has a problem, where the problem is. And similarly is the case for the molecular machines. But of course, we have a problem of size. So if we think about the exosome, this is just a core complex, only the core 10 proteins, which I will tell you about. And you think just in terms of dimensions, I mean, the numbers that you see here on this slide, I cannot even pronounce them. They are kind of staggering. So in terms of size, say it's about 10,000 of the thickness of your hair. In terms of weight, it's billions of times lighter than your hair. So clearly, to be able to see these machines and to be able to see them at atomic resolution, which is what we would like to do, we have to, we cannot just see them by eye. We just have to use specific methods. And over the years, many years, these methods, as it turns out, come from physics. So originally, we have been using methods based on X-rays. In fact, we have been coming to Hamburg, to the DAISY site, to the synchrotrons for many years. We have recently moved to use electrons, electron microscopy, and of course we are now trying to incorporate the information that can come from machine learning approaches. So this core complex, which I will tell you about, which I've been interested for many years, recognizes the free 3' end of RNAs, in fact, not just messenger RNA, but lots of RNAs, and degrades them one nucleotide at a time. And this complex has been conserved through evolution. In fact, when I started my lab, I started working on a very archaeal form of this complex, in fact, from archaea. But for the past, say, 20 years, we have been focused on eukaryotes. So it's been It's present in all kingdoms of life, is highly conserved. And what we've been doing in the past really 15, 20 years is to focus on 2 species. And we focus on 2 species. We focus on Cerevisiae, budding yeast, and on the human complexes. And we like to study these machines from these 2 species because it allows us to address different type of questions. So it allows to look at what are the conserved fundamental principles that have been conserved between these 2 species, which diverged more than 1 billion years ago. So there must be a core, really fundamental core of chemistry, which has been conserved through this long evolutionary distance. But also, of course, we want to understand the complex layers of regulation that exist in multicellular organisms, and particularly in the human case, of course, we have the context, disease context. So what does this machinery look like? One way of representing it, and this is now budding yeast, these are the proteins, and the names are not important, but what I try to do here is to color them in layers, because the way we think about this machine is really in forms of layers. And the layers are centered on a core. This is the catalytic core, and has another layer of factors, which are nuclear and cytoplasmic, and there is another layer of enzymatic activities. These are RNA helicases, I will tell you about it, and there is another layer of regulators. Now, I like this picture because it kind of shows the way we think and how it works, but it's very misleading, and the reason it's misleading is that these molecules are drawn with the same blob, okay? So they're not on scale. And really to try and understand the pathway, you have to think of this molecule in terms of really their physical size and what they look like. Because some will be, small folded proteins. Other will be like a spaghetti, and the way they work is very different. So this is in human, in yeast. In human, very similar, okay? There are some differences. I'm not going to go through the details here, but it's kind of similar, but there are more regulators. There are more yellow proteins, and there are even more regulators on the way these machineries are recruited on features that are present on the messages, on these transcripts. Now, in humans, of course, we can look at the context also of disease. And many of these subunits have been identified in patients with rare disorders. And they cluster in different types. So we have different types of diseases that are connected to these different types of mutations in these different aspects. And perhaps it's not so surprising if one thinks of the number of transcripts and the variety of transcripts that this machinery deals with. And in fact, it essentially is like I don't know how to call it, the Hoover of the cell kind of thing. It just deals with a lot of different transcripts, both in the nucleus, actually, where a lot of the action happens, and in the cytoplasm. And there are many different pathways where it degrades RNAs that are due to aborted transcription initiation, turnover. It's also involved in processing and maturation. I will not get to that today. And in the cytoplasm, is involved in co-translational surveillance. So this is messenger RNAs that have a problem, and the problem is decoded in the context of the ribosome. And what happens, most often than not, is that there is an endonucleolytic cleavage near the ribosome, which is the entry point for these machineries to come in and degrade the body of the transcripts. And this is one particular type of disease, which will tell you, which is connected to this type of regulators. It is connected to, this is a syndrome, it's intractable diarrhea. It affects actually multiple body systems, not just the gastrointestinal tract, but multiple body systems. How it works is unclear. And the mutations are in the yellow sphere, okay? It's the regulators. This is where the mutations are. But to tell you about the regulators, I have to start telling you the inner core to be able to then proceed to the rest. So now this is let me just see if I can make it go. Yeah. So this is now, the structure is a cryo-EM structure it's a structure from a cryo-EM data of the human cytoplasmic exosome core. So what you see at the bottom in pink is the RNAs. This is where the reaction takes place. The reaction is highly controlled. The RNA has to be threaded into this gray orange cage to be able to reach the ribonuclease. And to get there, it has to be threaded by this blue molecule. This is a helicase. And if now we look at the yeast structure, again, 1 billion year of divergence, they're very, very similar. So the mechanisms with which the RNA, and I'm not going to go through the details, so the chemistry with which the RNA is degraded at the bottom is the same. The mechanism with which the RNA is recognized by this inert cage has been maintained through evolution. The mechanism with which the helicase unwinds, substrates the RNA and threads it into the cage and then to the ribonuclease is conserved. So this is the mechanism so we have a helicase, it will recognize the substrate, which is a major issue, which I will get to, unwinds and threads it to this, channels it to the degradation chamber. Now the helicase is transient. What you see here at the bottom is actually a constitutive stable complex in the cells, and so the helicase will bind transiently to the stable complex to degrade the substrate. So the bottom is the machine, is the shredder. The helicase is the hand up here, which threads the substrate inside. And as you imagine, the hand is connected to the body. So a lot of regulation is in the hand. How is the hand recognizing the right substrates is a major point of regulation. And so work over many years, we've now understand it. Now this is in human, but in yeast is the same actually. The blue molecule that you see here, so this is the helicase. Actually the density here, this is the actual density, is the actual data, also in the previous slide. It's coming into the helicase. This is the engine, the ATPase. There is also this part of the molecule, I call it the arch domain, I will get back to it. And this is embedded in a complex, again, it's a constitutive complex in cells where you have these subunits over here, these are regulatory subunits. They have no catalytic activity. And this is a kind of scheme of how it works. And when this helicase is a part of the complex and binds RNA, the RNA is blocked, it cannot exit. Okay, and it cannot exit in this conformation because we have this part of the molecule, the yellow and green part, which blocks the bottom. We call it the gatekeeping module. And so what has to happen, and this is again a mechanism that is conserved from yeast to human, is that the helicase has to detach from the rest of the complex. It is still connected, okay, because there is this part, this unstructured part at the N-terminus, which forms the hydrophobic core of the yellow protein, but it has to detach for the RNA to be able to get out. So now we have the structure. We know exactly how this looks like. So of course we can try and see, well, where are these disease mutations in these patients? And some are very easy to understand, okay? Because for example, in this case, actually this is here the RNA density. This is, you see, the 3' end has entered. It's not proceeding because you have the rest of the molecule here blocking it. And here are, for example, 2 sites of disease mutations. And as you see, it's very near the RNA, it's near the active center. And so what we can do is to reconstitute the complexes biochemically and to both in the wild type form and in a mutated form and ask, does it affect the activity as you would expect? And in fact it does. This is the wild type and this is now the one of these 2 mutations here. And you see this, what we're looking here is an ATPase assay. This is an RNA-dependent ATPase, and essentially the activity is almost the same as killing the active site residues, the actual catalytic residues. There is another point here in this curve, which I will come back to, that if you remove this arch, which I showed you, actually you increase the activity. So this is a point of regulation. And actually it's another point where we have disease mutants. So now I'm looking more globally, and we are now plotting all the disease mutations. And as you see, we have some, I showed you, they are near the active sites, so clearly they have a role in killing the activity. Some of them are over here. They are in the hydrophobic core of the complex, and so what we would expect is essentially it affects the folding of these molecules. But there are some mutations that we couldn't understand, and one is here at one side of the molecule, on one of these long scaffold proteins, and the other one is around this arch domain that I showed you here. And for us, actually looking at this, where these disease mutations are, often gives us a hint to pay attention of part of the molecules that we may not necessarily pay attention to, because if there are things like here that we don't know what they're doing, it means that we don't understand. There is some aspects of this molecule that we do not understand. And so for us, in a way, disease mutation is a way to look at the interesting parts of these molecules. So, and in fact, this part here, we know what it does, and this is work we've been done a few years ago, at the time in collaboration actually with Roland Beckman. And the arch domain here of the helicase, in fact, interacts with ribosomes. And I'm going to show you in a while, so in the next slide. So we know this interacts with it's a major ribosome interacting site. What we know from data from the Pestov and the Buller lab, we know that actually in human cells, this molecule, this ATPase, can extract the message from stalled ribosomes, and the main function is in this quality control, co-translational quality control pathway I showed you in the first slides. So what does it do? Well, of course, we can reconstitute. In this case, these are the biochemical reconstitutions where we purify ribosomal subunits, we make all the proteins that we need to reconstitute this complex, and now this is a composite cryo-EM map catching the ribosome on the exosome. The way we did it is biochemically, we kill the ribonuclease activity, we have a single active site mutation, and we have an active helicase. And what we are trying to do with the system, have a system where the helicase is extracting the mRNA or the mRNA mimic from the ribosome, it threads it to the ribonuclease, but it cannot let go because the ribonuclease is dead. And there were two sides of the how is this here? Wait a sec. Okay. Go away. Do you see this thing? No, I only see it. Okay. Okay, so if I now run it, how does it work here? Weiter. Okay. You see here, this is a composite cryo-EM map. Here you see the helicase, you see the arch domain contacting the 40S subunit of the ribosome, and you see how we expected the mRNA. Actually, you don't see it. It's encaged in the ribonuclease complex. But we also saw density in this part of the molecule that we did not expect, and that's this gatekeeping site of the molecule. And if we now look, these are actually exactly these points where we had these disease mutations that we didn't understand about. One was in the arch domain, which is the main interacting site, but the other mutation, which I showed you at the end of the molecule, are here. And this is a side of the molecule which is very interesting, and the reason it's interesting is that there have been data recently on what you call colliding ribosomes. Actually, the first person, in fact, to visualize it is Daniel Wilson, who is here now in Hamburg. And these collided diazomes form a particular interface, which is the site of interaction. And what you see here in color is our structure. In gray is a structure of a diazomes that we have just superposed, and it just sits right there. And so the hypothesis is that, in fact, this regulator, is in fact the part that recognizes this ribosome collision, which is a sign of problems in the cells. And we have validated biochemically, and we are currently trying to push it to try and understand structurally. So over the years, essentially we have all these snapshots, okay? And I just showed you a few on the way. And so what we can do is to get to very high resolution, but it's just a snapshot. And so we have to try to think, how do we connect it, to try and think what is the process, because we do not have dynamic information in this high resolution, in the structures I showed you. And so what we have done is to work with Janet Ivaza and Margot Riggi, and we have tried to collaborate with them to try and put in a molecular animation to see how it could work. So now this is fiction, which I'm going to show you, but it's the way we think the process works. There are things that probably are not going to be correct, and there are things that, as we made the movie, we realized we don't understand. And I'm going to tell you on the way as we show it. So you have a ribosome. The polypeptide chain is coming. And here there is a problem. The ribosome stalls, and there is a colliding ribosomes that comes here. Now, there has to be in these pathways a 3' end cleavage, an endonucleolytic cleavage. And what we think is happening is that this key complex comes, the helicase binds, it threads the RNA, the gatekeeping module is a disome structure, is at the disome interface, and this is going to then recruit the exosome core. This is a transient interaction, as I showed you, and this is what's then degrade the message. Now, there are things we don't understand. We don't understand what is the relationship with the polypeptide chain, which I conveniently didn't talk about, so we don't know when this is happening. We don't understand whether these machineries are scanning. You know, it's present in much lower abundance than the ribosomes. Are they scanning? And then they stop when there is a problem. And we don't understand how does it interface with other quality control pathways and ribosome quality control pathways in the cell. And so just the process of making these movies highlights for us the questions that we need to address next. So what do we want to do next? Well, obviously, we want to try and understand how this complex, we want to visualize it onto a problematic disomes to try and see how it recruits the exosome there. Turns out we are probably missing some components, and in fact, there are other factors that have been recently identified which also are mutated in genetic disorders that we believe are part of the recognition systems. And so obviously, this is what we are going next. We have to increase the complexity of the biochemical reconstitution to be able to target this particular disome information. And this is what I would like to do with the generous funding from the Jung Foundation. And with this, I'm at the end of the talk. And I would like to acknowledge the many people who have worked on this. We have worked on this for many years, and you see the upper layer. Actually, we've worked also on the nuclear part. Today, I only touched on the cytoplasmic side of the exosome. And many of these talented students and postdocs now are have their own lab, and they're following their own dreams. And these are at the bottom: Achim, Yazemi, Chris, Fabienne, Jerome. Also together, the part of today was with Eva, Alex, and Ingmar, are really pushing the next questions in the lab. I'm very grateful to our facilities, crystallization, cryo-EM, mass spec, Margot for the animation. I'm very grateful to funding, and I'm grateful to your attention. Thank you.